All right, here we go. We're going to finish up the immune system now. We're going to talk about the adaptive branch of your immune system. This one may take a little bit longer. The adaptive immune system is more complex than your first and second lines that make up that innate branch of your immune system. But we're just going to kind of hit some highlights here. We're not going to get into a lot of gory details. So again, the microbes that are good at causing disease have evolved ways to get past your barriers, your skin and your mucosal membranes, and also to avoid those second lines of defense that we talked about. They're good at avoiding macrophages when they first invade your body. They're good at avoiding neutrophils. You'll learn more about why that is when you take microbiology. And for those types of invading microbes, and some others as well that may slip through the cracks, this third line of defense has to kick in. And this is called the adaptive branch of the immune system. Some people call it the acquired immunity, because you only develop this after exposure to a particular microbe. Those other things we talked about, inflammation, neutrophils detecting bacteria and swallowing them up, those types of things happen immediately. You don't need prior exposure to a particular microbe for those to kick in. Now, we mainly think about the immune system protecting us from infectious microbes, but it also protects us against abnormal body cells as well, like cancerous cells. It will also play a role in things like tissue rejection as well. Also plays a role in allergies. That's a special case where this particular branch of your immune system is overreacting to. Well, it's actually, allergies involve multiple branches of the immune system, but it's an overreaction to something that you are exposed to. And you'll hear where it says activates complement. Complement is a protein-based set of weaponry that your immune system uses. We're not going to go into the details of that here. You will hear more about that when you take microbiology. Some key features of your adaptive immune responses, very specific for particular types of microbes. You get exposed to this year's strain of the flu. The immunity that you develop from that probably isn't going to work very well against next year's strain of the flu. It's very, very, very specific. It's systemic, so it's not just localized. Like inflammation can be a very localized response. An adaptive immune response, once it is stimulated, extends all throughout the body. And then this is key here too, memory. This branch of your immune system remembers that you've been exposed to a particular microbe, and that allows it to respond much, much, much faster the next time. There are a couple of branches of the adaptive immune system. One is called humoral immunity. Humoral here is referring to body fluids, body humors. That's an old term from way, 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 way back in the day for body fluids. This is referring to antibody immunity. And you've probably already talked about antibodies when you did your blood typing lab. Antibodies are protein weapons. They're not cells, they're not white blood cells. They're made by a type of lymphocyte, but they are really proteins that float around in your body fluids, and they're supposed to serve as little tags when they attach to something that lets your immune system know, hey, this is something that's foreign and we need to get rid of it. That's the humoral branch that deals with antibodies. Then you also have what's called cell-mediated immunity or cellular immunity, which involves cells whose job it is to help detect and eliminate invading microbes in a specific way. Okay, another key feature of this branch of your immune system is that it reacts to antigens. Hopefully you've already talked about antigens a little bit in your in the laboratory part of the course. Okay, so I'm just going to draw what is an antigen. Let's say this is a bacterium that has invaded your body. Just like our cells, the cells of a bacterium or the cells of a yeast or whatever that have invaded the body, or the cells of a red blood cell from a mismatched blood donor, these are going to have molecules on their surface that are foreign to our bodies. Okay, so this is a little bacterium here. I am grossly over-exaggerating the sizes of these molecules. Many of these are proteins. They can also be carbohydrates that are located on the surfaces of these cells. Also on viruses as well. Viruses have antigens on their surfaces also. So they're just molecules. They're big, large, organic molecules. And your adaptive immune system sees these as foreign to you. So it's some kind of surface protein or carbohydrate that you don't produce. Your body doesn't make this. Your immune system is not supposed to react to things that are normally present on the surfaces of your cells. It's only supposed to react to things that are foreign to your body. Ideally, sometimes the immune system screws up. We'll talk a little bit about that toward the end of the lecture. Okay, I drew some of these surface molecules on this little bacterium that's invading your body with different shapes, and that's because a single bacterium is the singular form of bacteria, can have many different types of antigen molecules on its surface that it can respond to separately. Antigen, that word literally means antibody generator. That's where it got its name from. Stimulates your immune system to make antibodies. So keep that in mind, what antigens are. Cells of your adaptive immune system. The key players are these guys. Your B lymphocytes or your B cells, your T lymphocytes or your T cells. B cells are primarily involved in humoral immunity or making antibodies. Antibodies bind to antigens. Okay, keep that in mind. So back here on our little diagram of the bacterium, your immune system makes antibodies, which often have kind of a Y shape, like I'm drawing here, that bind to, this is an antibody, and it's binding to this antigen. And by doing that, these will coat this foreign invader in your body and tag it. If something in your body has antibodies that are coating it, that lets your immune system know, hey, this is something foreign to the body and we need to get rid of it. It will stimulate those phagocytic cells to come in. They're like maggots. They're like magnets for those types of cells to come in, hey, attach to this, gobble it up, and destroy it. You also have cells that are called antigen-presenting cells. Those are macrophages and dendritic cells, another role for macrophages, aside from just gobbling things up and digesting them. We're not going to talk about that, though here you will hear about that in your microbiology class. But let's talk a little bit more about lymphocytes too, and how they respond to these foreign invaders. So you've already learned that all of your blood cells originate in your red bone marrow. B cells mature there. T cells leave the bone marrow in an immature state, and they mature in the thymus. Now, where do these lymphocytes go after they're mature? Yes, you have some of them that are floating around in your blood because you've seen them hopefully in lab on the blood smears. But many of these cells lurk in your lymph nodes. Those are great places for them to hang out. You have them in your spleen. You have them in those malt tissues like your tonsils and your Peyer's patches. And they're hanging out in these locations because those are great places for them to intercept invading microbes. You know, things that you're breathing in or that you're swallowing from your food and drinks, or in the lymph nodes, they're going to intercept invading microbes that are draining off of your tissues with the lymph fluids. All right, so let's see what happens here. We're talking about, more specifically, your B cells, or B lymphocytes. Those mean the same thing. Okay, here's the deal with B cells and T cells. Your red bone marrow generates B cells and T cells, an almost unlimited number of them, that can bind to an almost unlimited number of different kinds of antigens that your body gets exposed to. There are literally billions of possibilities. But one B cell will recognize and bind to one antigen. Say like a molecule on the surface of influenza H1N1, that would be an antigen. Or a molecule on the surface of a Salmonella bacterium. A protein molecule on the surface of a pollen grain. Those are all examples of antigens. A B cell that binds to a protein on a pollen grain is not going to bind to a protein on the surface of an H1N1 influenza virus. They're very, very specific about what they bind to, but your red bone marrow cranks out B cells and T cells that can recognize an almost unlimited number of different types of antigens that you get exposed to. What they're showing you here in this diagram, these are supposed to be 3 different B cells that are probably hanging out in a lymph node or in a tonsil somewhere, and along comes, they're showing this as an antigen. We'll say that's a bacterium of some type, which is covered in antigens on its surface, and this one little B cell gets lucky. The Y-shaped molecules on the surface that kind of look like the antibodies I just drew, those are receptors on the surface of the B cell that bind to a particular antigen. So all the receptors of this B cell bind this particular antigen. The receptors of this one and this one don't. These guys aren't going to do anything. They're going to stay in a quiet, unactivated state. This one, though, now there are some other things that have to happen in order for full activation to occur in most cases, but we're not going to go into all those details. So for our purposes, when a B cell gets lucky, sees its antigen, it becomes stimulated to divide. That one cell divides over and over and over again. Each of those offspring are clones of the original cell. They're all programmed to respond to that same antigen that cell bound to up here. So these cells divide over and over and over again. You'll have millions and millions and millions of copies. A lot of times this is happening in the lymph node. Your lymph nodes may be swelling because this is happening. And most of those cells are going to specialize into a special type of B cell called a plasma cell. This is like a mature B cell. Okay, and this is a different use of the term plasma. These cells, their job is to secrete antibodies. These antibodies will bind to these antigens up here at the beginning that stimulated that original single cell. They bind to that same antigen, so keep that in mind as well. So now you're going to be, these cells are going to be secreting tons and tons and tons of these antibody molecules, which get released into your lymphatic fluids. Those end up in the bloodstream. They get returned to the subclavian veins. They enter your blood circulation. They're going to flow to all tissues in the body, and they can exit those tissues in locations where you have an infection occurring and bind to these little microbes that have invaded the body. They basically tag them. Here's a bacterium. It gets antibodies that are covering it. Those antibodies function like little tags that tell your macrophages, for example, get over here and bind this guy and digest him and destroy him. He's foreign and we need to get rid of him. Okay, now on this diagram, I said just a second ago, this is the one B cell that got activated. It divided over and over again. Most of those clones that came from that single cell become these plasma cells that make antibodies. I said most. A smaller percentage of them specialize and become, I'm going to erase that, they become these guys. These are called memory B cells. What memory B cells do is they don't immediately start making antibodies. Instead, what they do is they stay in your lymph nodes, they go to your tonsils, they go to those malt tissues down in your intestinal tract, and they just kind of hang out there and lurk there, and they can live for many, many, many years. And then if you get exposed to the same invading foreign microbe again, and these cells bind to an antigen on the surface of that invading microbe, that same antigen that we were looking at up here, what happens is these cells, these memory cells, divide very quickly, much more quickly than the first time up here. They very quickly multiply, make lots and lots of plasma cells and more memory cells, and these plasma cells will secrete lots and lots and lots and lots of antibodies. This is called a secondary response, the second time you're exposed to a particular microbe. Again, it's very, very, very specific. So this is called, again, this is really what your immunity is. This is immunity due to memory. And so this is how vaccines work. When you receive a vaccine, you're receiving antigens in an artificial manner in order to generate these guys. You want to make these memory B cells. You can also make memory T cells, but you'll hear more about that when you take microbiology. And then these guys lurk in your lymph nodes, your tonsils, etc. And then if you ever really do get exposed to the real microbe that can make you ill, those memory cells will respond so quickly that usually the antibodies that are produced will intercept these microbes before they can multiply enough to make you sick. That's the basis for how vaccines work. And again, this is much, much, much faster than this process up here. The first time it happens, it can take 1 or 2 weeks before you start making antibodies. Down here, it can take just a couple of days, very, very quick. All right, so again, humoral immunity. This branch of your immune system deals with making these antibodies, these protein weapons that bind to foreign invaders. I do want to talk about some different categories of immunity that you can have. Humoral immunity, antibody immunity. You can have what's called active immunity, and you can have what's called passive immunity. Active immunity means your immune system, specifically B cells, make antibodies. That's active immunity. Passive immunity means you receive pre-made antibodies. Okay, now under active immunity, there are two categories of that. Naturally acquired. This is what we've just been talking about. This is where you get exposed to a particular infectious microbe. You have an infection, you've come into contact with a pathogen, that's a disease-causing microbe. Your immune system responds and you make antibodies that will bind to that microbe, tag it, and help your immune system get rid of it. All right, that's called naturally acquired active immunity. Artificially acquired active immunity is what I was just talking to you about with vaccines. When you receive a vaccine, that's a preventative, and the idea there is let's inject some antigens of this microbe into the person. And sometimes that may be, sometimes you may actually receive a live virus or a live bacterium, but it's weakened in a weakened state, like if you've ever had the intranasal flu vaccine. But whatever the case, the idea there is to deliberately expose your immune system to the antigens of some microbe to stimulate B cells to make antibodies to it, and also to stimulate the development of memory. So when you really do get exposed to this particular microbe, you'll be able to rapidly produce antibodies to it. That's called artificially acquired active immunity, and again, your immune system is making the antibodies. Passive immunity, this is where you receive pre-made antibodies. And this can occur naturally. You've probably heard before that a mother's breast milk contains antibodies made by her immune system, and those can be delivered to the infant through the breast milk. The infant will absorb some of these antibodies in the breast milk through the intestinal lining. When you are a newborn, you don't have a lot of adaptive immunity built up yet. You haven't been exposed to a lot of things yet. You don't have any memory that allows you to quickly react to invading microbes. So these antibodies from mother's milk can help a newborn infant until his or her immune system develops better. Also, antibodies can pass across the placenta from mother to fetus in some cases. Artificially acquired passive immunity, this is not really so much like a vaccine. A vaccine is stimulating your immune system to make its own antibodies, but there are cases where you have to receive pre-made antibodies. They were already generated and purified, highly purified. This occurs in cases like if you have a snake bite. You've been bitten by a king cobra, you were over in India, were bitten by a king cobra, and they're going to give you an anti-venom, or sometimes those are called antivenins. What that is, those are pre-made antibodies that have been highly purified, and they bind to the snake venom or spider venom or whatever it is. That's functioning as an antigen that these antibodies bind to. So you don't have time. You get bitten by a king cobra, you don't have time to wait around for your own immune system to make its own antibodies. You need some pre-made antibodies, and you receive those artificially. That's what you're getting when you receive an antivenin or antivenom. And that's called artificially acquired passive immunity. It's not a natural process, it's an artificial process as part of a medical procedure. So y'all, just a little bit more about what these antibodies are. They're also called immunoglobulins. That's another term for antibodies, means the same thing. And again, they're proteins. They're secreted by these plasma cells. Those are activated B cells. They bind to very, very specific antigens, and they tag them so that those, whatever they bind to, will be destroyed by other parts of your immune system, usually macrophages, neutrophils, etc. Those cells whose job it is to bind invading foreign things in the body and digest them. Antibodies are Y-shaped molecules, and this is supposed to be trying to give you a little bit more of a feel for the three-dimensional shape. One Y-shaped antibody molecule is actually built from 4 proteins. Here's one, here's a bigger one, number 2, here's another one of those bigger ones, number 3, and there's number 4. We're not going to get into a lot of the nitty-gritty details here. The really important kind of business portion of an antibody is right here at the tips of the Y. These are your antigen binding sites, right there and right there. So a particular type of antibody that binds to a particular antigen, like if my fist was an antigen, the antibody that binds that little pocket there is going to perfectly fit my fist, that antigen. That little pocket is not going to perfectly latch on to another type of antigen that has a different three-dimensional shape. So the different antibodies that we produce along the tips there, those antigen binding sites, that's really where they vary. The rest of the antibody molecule looks the same from one nut type to another. It's those tips of the Y where you have that specificity that allows them to bind to particular antigens. All right, we're not going to talk a whole lot about cell-mediated immunity. Cell-mediated immunity is a phrase that's generally used to describe T-cell immunity, the function of these T lymphocytes. T lymphocytes fall into 2 major groups. Let me write this down here. T lymphocytes, you have 2 major groups. You have helper T cells, or TH cells, and you have cytotoxic T cells, or TC cells. We're mainly going to take a quick look at cytotoxic T cells and what they do. The helper T cells, as their name suggests, they help other types of cells that are part of your immune system. They're sometimes called the grand conductors of your immune system. They're very important, but their functions are pretty complex, and we're not going to talk about those here. You'll hear more about those in micro. But let's talk about these cytotoxic T cells, because they are a very important branch of your immune system. And one thing that's key here, the targets: virus-infected cells, cells with intracellular bacteria or parasites, cancer cells, foreign cells like cells that you would receive from a donor in a blood transplant or a tissue transplant. The key thing here is these guys, cytotoxic T cells, they kill your cells in most cases, or they might kill transplanted or transfused cells. Why on earth would we want to kill our own cells? Here's the key. Viruses are microbes that don't, well, let's back up here for a minute. When you are infected with bacteria, very often those bacteria multiply in the extracellular spaces, in the extracellular matrix between your cells. They don't always, not all bacteria go inside your cells, they stay out there. Hey, that's great. So you make antibodies. Antibodies can float into these tissue fluids from your bloodstream, and they can bind those bacteria that are hanging out there in your tissue spaces. That's great. They're tagged for destruction, and now macrophages are going to come in and swoop those up and gobble them up. That's wonderful. Viruses, though, are a little more sinister. Viruses multiply inside your cells. They don't multiply in your tissue spaces. Viruses have to go inside your cells to multiply. Same thing, there are some types of bacteria that do this as well. They're called intracellular bacteria or intracellular You can have intracellular parasites as well, like malaria parasites, for example, are a type of intracellular parasite. Well, guess what? Antibodies don't go inside your cells. Or they don't do that very efficiently. So viruses and intracellular bacteria and other parasites, they're basically hiding from antibodies. And if we're going to get rid of them, the only way for your immune system to get rid of them is to kill the cells that are infected with them. And that's where these cytotoxic T cells come into play. They make a lethal hit. And we'll see there, I've got a diagram on the next slide which will show us how that all works, and so I'll explain that on that slide. I'll tell you what, guys, just let's leave this out for now. You can read about that in your textbook, or you may hear more about that in your microbiology class. Let's take a look at what's going on here. We have a cytotoxic T cell, and it has migrated into one of your tissues somewhere. This is its target cell. So this is one of your cells, probably infected, let's say with a virus in here. So what this cytotoxic T cell does is it's able to recognize your cells that are infected with something. And they're very, very specific about what they recognize. So let's say this cytotoxic T cell right here is programmed to recognize your cells that are infected with influenza virus. It's not going to recognize one of your cells that's infected with hepatitis B virus. It's not going to recognize one of your cells that's infected with human rhinovirus that causes the common cold. Like antibodies, these cells are very, very specific about what they bind. That's why they're part of your adaptive defenses. These kinds of cells don't get activated in the first place until you become exposed to a particular invading microbe. Okay. How it recognizes them, you'll learn more about in your micro class, but whenever one of your cells is infected with a virus or a bacterium or some sort of parasite, it actually takes some of the antigens of that virus or bacterium and displays them on the surface in membrane proteins. And that's the flag that calls over your cytotoxic T cell. Okay, so what happens here? You've got a cytotoxic T cell that has come together with one of your cells that's infected. These two cells come together, and that's what we're looking at here in this particular diagram. A couple of things that happens. The cytotoxic T cell releases proteins that are called perforins. Like perforate, they actually assemble and form a pore in the surface of the target cell, pokes a hole in the membrane. The cell also releases enzymes that are called granzymes that are represented by these little dots here, and those flow through those pores into the target cell. Well, I shouldn't have drawn that one because there was no pore there for it to flow through. What these enzymes do, these are enzymes that basically stimulate apoptosis, or they trigger the cell to commit cell suicide, to basically digest itself and die. It digests its own DNA and dies. The little perforins, these little pores that they form, these holes that they punch in the membrane, that's not good for a cell to have holes in its membrane because important molecules can leak out, water can rush in, and that's also going to help the cell to die. So the end result is your little target cell here that's infected with something, or this also works in some cases, it's not 100% effective, but these types of cells are also involved in detecting and killing some types of cancers, transplanted tissues, etc. These are like hitmen for your own cells. The idea there is, hey, let's go ahead and sacrifice some of our own cells to help us get rid of a virus or an intracellular bacterium that has caused an infection. Okay, so that's kind of a quick, just hitting the highlights overview of your adaptive immune system. A couple of other things before we go. Immunodeficiencies. The definition there is kind of self-explanatory. This is where one or more branches of your immune system has become deficient. It's not working properly. And there are types of immunodeficiencies that are congenital, so you're born with those. And then some are acquired. A really important example of that is AIDS. That stands for Acquired Immunodeficiency Syndrome, which is caused by HIV. And how that works is HIV kills helper T cells, as I mentioned, those are like the grand conductors in many ways of your immune system, and your whole immune system essentially collapses when you have full-blown AIDS and you cannot fight off infections. So there are different types of immunodeficiencies that can occur, and just be aware that those exist. Another thing you should be aware of is autoimmune diseases. Auto means self. Your immune system is not supposed to react to your own antigens. You have antigens, cell surface molecules, molecules in your tissues, etc. Those are antigens. They're not foreign antigens, but they're still antigens. Your immune system is not supposed to react to those, but there are cases when your immune system becomes defective, and it may start to react to one or more of your own antigens. You might make autoantibodies, antibodies that bind to one or more of your own types of antigens, or you might generate cytotoxic T cells that seek out and destroy some of your own cells as well. Various examples. Multiple sclerosis is thought to be an autoimmune type disease. Graves' disease. When you study the endocrine system, you will talk about Graves' disease. That's where you make antibodies to thyroid hormone receptors, or thyroid stimulating hormone receptors, on the surfaces of cells in your thyroid. Diabetes mellitus, type 1 diabetes. That is an autoimmune disease where you generate cytotoxic T cells that kill the cells in your pancreas that make insulin. Rheumatoid arthritis. This is where you're generating antibodies that bind and accumulate in your joint. So there are several different. Systemic lupus erythematosus, SLE, are just commonly referred to as lupus. You wind up making antibodies to many of your own antigens, and that causes a wide array of problems. One last type of immune system problem that you can have are called hypersensitivities. And many of these are what we refer to as allergies. There are different types of hypersensitivities that you'll learn more about in your microbiology course. In fact, there are 4 different types. And what we call type 1 hypersensitivity is what we generally refer to as kind of common allergies. This is basically where you have an overreaction of your immune system that can stimulate, in some cases, localized inflammation, or in severe cases, widespread inflammation stimulated all at once throughout your body. And a really extreme case of that can lead to anaphylactic shock. This is triggered. Let's use an example of like somebody with a peanut allergy. That means that your immune system has developed antibodies that react to one or more antigens on the peanuts that you're eating. Antigens that cause allergies are referred to as allergens. And again, these are things like proteins, carbohydrates. These are big molecules. Okay? So when somebody tells you they have an allergy to salt, or that's not an allergy. They're not really having an allergic reaction. You don't make antibodies to little tiny things like that. You make antibodies to large organic molecules, mainly proteins and carbohydrates. In cases where people develop anaphylactic shock due to an allergy, what happens is you have a widespread, all-at-once reaction to these allergens that have been absorbed into your bloodstream. And it's kind of like, instead of just localized inflammation, you have widespread inflammation all across your body all at one time. That leads to, now we just talked about how inflammation in a local area causes vasodilation. So you get the leaking of the plasma into the surrounding tissue fluids, you get the redness, you get the swelling. In this case, it's like you have inflammation that develops everywhere at once. You have blood vessels that are dilating everywhere in your body. So you have fluid that's lost from the bloodstream. Guess what happens to your blood pressure? You become hypotensive. Your blood pressure plummets. You can no longer circulate your blood properly to vital organs. And if it's not taken care of pretty quickly, you're gonna croak. Also, can stimulate constriction of the bronchioles in your lungs. Those little tiny air passages actually constrict and make it difficult for you to breathe. Like when somebody has an asthmatic type reaction. Asthma is a reaction to some type of allergen as well. Treatment, epinephrine. Epinephrine stimulates vasoconstriction, constricts the blood vessels, and tries to jack the blood pressure back up. Also will help stimulate more forceful contractions of the heart, if you remember your cardiac cycle, cardiac output lectures. So epinephrine hopefully will help restore blood pressure that's high enough to start circulating oxygenated blood back to your tissue so that all of your vital organs will survive. All right, so that's the end of our 2 video clips on the immune system. And if you have any questions, feel free to contact me. I'll be happy to answer those for you.